Hey future doctors, welcome to NEET Podcast, where I am providing you with rapid revision of NCERT Biology chapters in less than 10 minutes for the upcoming NEET exam. This is Dr. Sharun. Let's start. Biomolecules. Introduction to biomolecules. Definition. Organic molecules essential for life processes, found in all living organisms, proteins, carbohydrates, lipids, nucleic acids. Building block of life forms macromolecules. Types of biomolecules: primary metabolite, secondary metabolite. Primary metabolite, essential for growth, development, and reproduction. Example: amino acids, carbohydrates, lipids, proteins, nucleic acids. Secondary metabolite, not directly involved in growth and reproduction, often have ecological function. Examples: alkaloids, morphine, quinine, flavonoids, terpenoids, pigments, essential oils, etc. Carbohydrates, polysaccharides or sugars. Definition: organic molecules made up of carbon, hydrogen, and oxygen. General formula is CH2O whole N. Function: energy storage, structural support. Types of carbohydrates: monosaccharides, simple sugars, sweet, soluble. Glucose C6H12O6, primary energy source. Fructose, sweetest sugar. Galactose, found in milk. Disaccharides, two monosaccharides linked by glycosidic bond. Sucrose is glucose plus fructose. Lactose is glucose plus galactose. Maltose is glucose plus glucose. Polysaccharides, complex carbohydrates. Storage polysaccharides, starch in plants, glycogen in animals. Structural polysaccharide, cellulose, the major component of plant cell wall. Chitin, exoskeletons of insect found in fungi and arthropods. Proteins. Definition: polymer of amino acids. Elements present is carbon, hydrogen, oxygen, nitrogen, and sulfur. Peptide bonds connects the amino acids. Structure of protein: primary structure, linear sequence of amino acids. Secondary structure, alpha helix and beta pleated sheets. Tertiary structure, 3D structure determines the function. Quaternary structure, multiple polypeptide chains. Example: hemoglobin. Function of proteins: enzymes, biocatalyst. Amylase, pepsin, structural protein. Keratin in hair, collagen in skin and bone. Transport proteins: hemoglobin, oxygen transport, GLUT4, glucose transport. Hormonal protein: insulin, growth hormone. Antibodies, immune defense. Enzymes, biocatalysts. Definition: proteins that speed up the chemical reaction, highly specific, reusable, work at optimal temperature and optimal pH. Mechanism of action: lowering of activation energy. Properties of enzyme: active site, region where substrates bind. Lock and key model: substrate fits into the enzyme like a key in a lock. Induced fit model: enzyme slightly changes shape to fit the substrate. Factors affecting enzyme activity: temperature, enzyme works best at an optimum temperature. pH, each enzyme has an optimum pH of its function. Substrate concentration increases the reaction rate until saturation of the enzymes. Inhibitors: competitive inhibition, binds to the active site, blocks the substrate, competes with the substrate for the active site. Non-competitive inhibition, binds to site other than the active site, changes the enzyme shape. Lipids, fats, oils, steroids. Definition: hydrophobic organic molecules made of C, H, and O. Function: energy storage, insulation, cell membrane structure. Types of lipids: simple lipids, fat, oils, and waxes. Fatty acid plus glycerol forms triglycerides. Saturated fats, no double bonds, solid at room temperature. Unsaturated fats, double bonds are present, liquid at room temperature. Compound lipids are phospholipids, make up the cell membrane. Steroids, cholesterols, hormones are steroids. Nucleic acid, DNA and RNA. Definition: biomolecules that stores and transfers genetic information, made of nucleotides, sugar plus phosphate plus nitrogen bases. Elements present: carbon, hydrogen, oxygen, nitrogen, and phosphorus. Types of nucleic acids: DNA, RNA. Deoxyribonucleic acid, double stranded, helical genetic material. Sugar present: deoxyribose. Bases: adenine, thymine, cytosine, guanine. Functions: carries the genetic instruction. RNA, ribonucleic acid, single stranded, involved in protein synthesis. Sugar present is ribose. Bases: adenine, uracil, cytosine, guanine. Types of RNA: mRNA, tRNA, rRNA. mRNA carries the genetic code from DNA to ribosome. tRNA brings amino acids for protein synthesis. rRNA part of the ribosome structure. So that was Biomolecules Rapid Revision Audible format. You'll find the PDF down in the description box. I hope you found the podcast helpful. This is Dr. Sharun signing off.